It's a really awesome molecule. But you're absolutely right. For a computational chemistry company that has been developing enzymes for third parties, how do we bring this product to the market? What are the industries? What are the applications? How do we get this out there? It hasn't been easy. It has been maybe the biggest challenge we've faced so far.
Welcome to this week's episode of Leaders on a Mission. I'm your host, Simon Leach, and today we're going to be diving into the world of enzymes, those invisible powerhouses behind everything from medicine to detergents to breads to beer to sustainable chemicals, right? So my guest is Fátima Lucas, CEO and co-founder of ZYMVOL. ZYMVOL are a really interesting company. They're using AI and molecular modeling to revolutionize how enzymes are discovered and designed. And man, this is a niche podcast. All the people that listen to this, they know a fair bit about kind of this world. So today we're going to kind of explore, how ZYMVOL are really disrupting the biocatalysis space and the whole future of computational biology, what it takes to build a high-impact biotech business from the ground up. So we got loads to cover today, Maria. Thanks so much for coming on.
Thank you, Simon. It's really a pleasure. You don't know me very well, but I love talking about enzymes and how technology can actually change the world. So thank you so much for this.
Well, you're the perfect guest for the show. And but tell us a little bit around, just give me some context that led to you kind of founding ZYMVOL, as it were. What was the key kind of experience that you had gained at that time that you founded the company?
Well, it's been mostly by chance, as a lot of things actually happen in science and technology. I had been working, so I'm a computational chemist by training. I worked most of my life in the area of drug discovery, so developing and applying methodologies to discover novel drugs. Even though it was an interesting field, and it is an interesting field, to be totally honest, I never felt like I would change the world. I never felt that I was particularly bright or had any ability that would allow me to design the best next drug. So at some point when a scientist came to our lab and mentioned that they were evolving these enzymes through a technique called directed evolution, which is basically Darwin's evolution sped up in the lab. And they would end up with these nice enzymes that had interesting properties that were industrially relevant. And the big issue they had was we found these enzymes mostly by chance because they're mutating the enzymes and once in a while there's a mutation that works. And they came to us and they say, we find these enzymes, but we have no idea why they're better. Do you think with your technology, your molecular modeling, you're actually able to tell and understand and characterize these enzymes, why they're working better? And to be honest, I'm a chemist. I grew up in a small village in Portugal where chemistry is overall present and all the contamination that comes with it. And suddenly I found myself in a situation where we could actually understand enzymes in the computer. But more importantly, and this was the evolution that happened in the first 3 years, is that we could actually design them. We could redesign them for specific industrial applications. And that's how I ended up in this field. And suddenly money was over. I was a scientist. There was no more, the project was done. There was no more resources at the department where I was working. And I found myself without a job. So my husband is from Barcelona. He wanted to stick around because of a nice job offer. And I said, well, I need to reinvent myself. And I basically just said, I think there's a market for enzyme design. Let's see if I can do it. And myself with the other two co-founders, we put some cash into the company. And after a few months, we were selling. And that's how it all started.
Amazing. Yeah, absolutely. So you bootstrapped, you put your own funds in.
We bootstrapped for 6 years.
Wow. that's some journey.
It is, and it was really interesting because as a scientist, I knew nothing about business. So here in Barcelona, there's a nice, it's an institution called Barcelona Activa that basically trains you in whatever field that you need. So I learned to put together a financial plan, how to do a market study. The first thing we did was reach out to more than 100 companies and ask, do you need enzymes? Are you using enzymes? How are you designing them today? Do you use any computational simulation? So I had to learn all of that by doing, and sure enough, we designed all of our products together with our customers. So we were very fortunate in that sense. And yes, there was a need for computational enzyme design.
Yeah, amazing. And what about that going from scientist to CEO, founder? Do you think at your heart, there was always this entrepreneurial kind of feel and flavor? Like if we were to go back 10 years or so and I would have said you're doing this now.
I don't think so. I don't think
Really?
I ever had the need, the intention of becoming an entrepreneur. What I can tell you today is that I wish I had done this 30 years ago, because everything that I've learned as an entrepreneur would have been extremely valuable as a scientist. But no, I don't think I ever thought about, like, I've seen many colleagues build their companies. It was never something that attracted me. But yeah, once you do it first time, I think you never get away from that entrepreneurial spirit, right?
Yeah, amazing. And so tell us a little bit about the mission behind, ZYMVOL, really, and particularly around how you envisage really disrupting this biocatalysis market.
So biocatalysis used to be something for the very rich. You were essentially a pharma company that had very deep pockets, and you were developing a novel drug, and you needed a synthetic route, and you could do this with biocatalysis, basically, because it's, at the end of the day, drugs need to be very pure. You need to have very clean products, and biocatalysis are great for that. So these companies were willing to invest a few million to develop a new chemical route, basically. Where I see biocatalysis going forward is that any company in the world doing anything can actually have this technology available. And that's where computation plays a big part, because if you do directed evolution in the lab, you're basically screening thousands, millions of enzyme variants in the lab to be able to find one or two that work. But this is time consuming. You need to have a high throughput method to be able to screen these enzymes in large scale. And of course, it's extremely expensive. By translating most of this work into a computer, we're basically screening these millions of enzymes, but with very good and realistic simulations that basically make you feel confident that whatever you send to the lab is very likely to succeed. And indeed, we do two things, discovery and engineering. But for instance, in the engineering part, when we engineer our enzymes in the computer, 90% of the time we will find hits in the lab. And we're talking about, instead of screening millions, we're talking about less than 100. Consider that a typical campaign for us, from early discovery to engineering, we send less than 300 enzymes to the lab. So this has a massive impact on time, but most importantly on cost. That means that now companies that are trying things for the first time, that are very skeptical about enzyme, they can do it because it's affordable. And even if they fail, it still doesn't bankrupt the company, but more importantly, because our technology is based on physics. So we have AI and physics. And I want to dig into this a little bit, because the physics based is like a flight simulator. So if you put in the right information into the flight simulator, you're going to crash. It's the same thing with enzymes. So we put in the information, and if there's no chances that you're going to succeed, don't put your money towards that development. Do something else. And this is, I think, the biggest value we give to our customers. It's not just disrupting the price and the timelines to develop enzymes. It's the fact that from day one, without even going to the lab, we're telling our customers, don't go this way. You're going to crash. Go this way instead, because chances of success are really high. And we've been doing this. It's been over 150 projects. And our success rate is between 80 and 90%. So we feel pretty confident when we're recommending an avenue for our customers. We feel pretty confident. And that's why I keep saying, mission, what do I want to do? Biocatalysis everywhere, clean chemistry. But sustainability is not the motor to take you towards biocatalysis. It's always an economic advantage. You need to have a process that will bring you, lower costs of production, less side products, less steps of purification. It always has to be an economic advantage. But for you to even get started, it needs to make sense. It needs to be cheap enough. It needs to be de-risked enough. Otherwise, people are not going to do it. A food company is not going to develop an enzyme, assuming that they're going to spend a couple of million and possibly fail. That's not going to work. So our mission at ZYMVOL, and what I'm really committed, is clean chemistry everywhere, for anywhere, for anyone, anytime. That's what we're trying to do.
Got it. Yeah. And are you finding that some of the drivers, because I don't know, across all sorts of industries, particularly in Europe, for instance, there are all sorts of regulations now on certain chemicals being banned. How much, is that just generally an opportunity for your platform and this technology?
Absolutely. It's all the molecules that are being banned, they need to be replaced by something else. Not only the molecule that is replacing needs to be sustainable, but the way you produce those molecules need to be sustainable. So think back a few years to COVID. A lot of the drugs that we use on our everyday basis, they're being manufactured elsewhere. It's not only the molecule itself being banned, it's how it's manufactured. If we want to be in Europe, independent of the rest of the world, we need to bring a lot of manufacturing back to Europe. And that means clean, sustainable processes. We have extremely strict regulation in Europe. You can't just produce anything in your backyard. And that's where enzymes really have a massive advantage. They can replace molecules that are being removed from the market, but they can also allow you to produce the new ones in a process that is sustainable. And it's not only that, and we often forget about this, but creation of jobs. Biocatalysis needs expertise, needs people that understand molecular biology, that understand fermentation. We're also creating jobs for scientists that are basically running away from Europe because there are just not enough jobs here. So the opportunities that are being created through biocatalysis is not just in replacing molecules that need to be removed. It's also in bringing this manufacture back to Europe and allowing us to be a little bit more independent, because today, at least in my view, we're way too dependent on the rest of the world, and Europe is quite isolated in that sense.
Yeah, and Europe has a really good history and heritage as it relates to this technology, right?
Absolutely. Half of the market is in Denmark. So biocatalysis, enzymes, Europe has always been the leader. But if we don't speed up, if we don't take this new wave, because computation is not here just as a fashion, a moment. It's not hype. It's here to stay. So AI, molecular modeling, all of these technologies are here to advance in biocatalysis. And in this particular area, United States and Asia are investing a lot. And I think, companies like ZYMVOL, and not only ZYMVOL, there's a lot of other companies here in Europe that are promoting biocatalysis thanks to simulations.
Yeah, absolutely. And it's always interesting looking at early stage kind of companies that are really leveraging kind of innovation and technology in terms of how they build the actual model itself. You get some, eventually they become product companies themselves and they try and bring something to market. But I think you're very clear about where you want to play, right? And your part to play in this.
We are very clear, but it is true that we are becoming ambitious. And even though we started with the early on value chain of enzyme discovery and enzyme engineering, we've already set up our lab. So for the past 2 years, we have a small lab that is able to do all the experimental validation. But we're also right now in a stage where we're partnering with different companies throughout the world so that we're able to help our customers scale up. And eventually, I think the business is in being able to deliver end-to-end solutions to your customers. So I wouldn't exclude the possibility a few years from now to be able to also produce enzymes, at least to a certain scale. Not saying we're going to be building 100,000 liter fermenters, but I think to a certain scale, it's important that we have that independence.
Awesome. Yeah, no, absolutely. And just so I understand this, right, because I'm no biologist or computational chemist. When you do the simulation, I'm interested in the process from, you've mentioned about you might send 300 kind of samples to the lab. But how many, when you're looking at this AI model, this physics-based kind of system, as it were, I'm interested in a couple of things. The context of how broad that search is, number one, and what it uncovers. And then, when you do get these hits, how does it translate in the lab? And how does that work? And then what happens from there?
Certainly, Simon. So it all starts with a large library of possible sequences. And by large, I mean a couple 100 million. And depending on the libraries, there are some private libraries out there that are billion sized. Okay, so we're talking about many potential sequences to search from. And this is just the search part, because then once you engineer, if you have a sequence, say 300 amino acids, each one of these amino acids can be replaced by 20 different types of amino acids. So the combinations, there's more combinations there than all of the atoms in the universe. Okay, so the scale is really gigantic. We use AI mostly to filter down this massive space into a smaller number of sequences to screen. Okay, so AI is helping us narrow down the universe of possibilities that we want to screen for. The physics-based is a whole different story. The physics-based is actually looking into the 3D structure of the protein. So we need to go from sequence to 3D model. And then this 3D model, we need to see how it interacts with the substrate, how when, everything moves, because you need to take into consideration dynamics, and how that substrate actually fits into the active site. So this is pretty complex to do in a computer, but this technology, it's coming from drug discovery. Okay, so drug discovery, I mentioned at the beginning, it's basically you have a protein and you have a small molecule, right? So you're just looking for millions of small molecules that could fit into this target. We're doing it the other way around. We have the small molecule, we know what the substrate is, and what we're looking for are which are the proteins that actually fit best and have the right chemical groups to be able to promote catalysis. So this is the computational part. We start with millions, we screen down to hundreds of thousands, and then finally we have a shorter list, which is in the hundreds. And here is where a lot of the magic happens, because even though computers are really good at doing their work, the input of scientists looking at these final hits is also extremely important. And I'm not saying that in the future you can't do everything without human intervention. I believe it will be possible once the AI models are more sophisticated. But today, at least at ZYMVOL, it still depends a lot on the chemical intuition of the scientists on the other hand. So now you have the sequences. These are normally between 20 and 90. Okay, so 20 sequences that are sent to the lab. At this stage, you need to order the gene. You need to synthesize it. You need to produce the enzyme. You need to then also buy the chemicals, reactants, products. So you need to put the enzyme together with the reactant, see if there's a change in color, which is the most typical way to see if there's any activity or not, and then just use a diverse options of analytics to actually confirm that you're producing the right compound. So it's not only that you're producing the right compound, is how quickly you're producing it, how pure is it, do you have any side products? All of this needs to be measured, and the final reaction, if you're lucky, is I'm able to produce a single molecule and I'm able to convert all of the reactants into that single molecule. So that's the best case scenario. So think about all of the fields that went into all of this, okay? Physics, chemistry, biology, molecular biology, fermentation. So there's just so many fields to be able to go from idea to, okay, now I can produce this with an enzyme, that it's extremely difficult actually to navigate all of this. And again, think about the customers that have never used biocatalysis before. It's a real nightmare, eh? For them, most of the time they don't understand exactly what we mean when we're doing this in the computer and then sending it to the lab. It's difficult to explain it to a customer.
Yeah. And how long does that process take from when you're dealing with the 20 to 90 and you're doing all the testing?
So typical project, it takes us about a month to do the computational work, and then it takes us about 2 months to do the lab work. So say 3 months, you have a first answer to your problem.
You've got a first answer to that problem.
Which is not necessarily the final answer. So then you might need to do more iterations of engineering to be able to get to your final product. But a typical project with us takes about 2 to 3 rounds. So in 9 to 12 months, you normally have an industry-ready enzyme. Then you still need to scale it up, eh? So we're only talking about the beginning of enzyme development. It works in the lab, but now you still need to produce kilogram scale, ton scale of this enzyme. Think another year to develop this.
Right.
Okay.
It's not a short-term project.
No, it is not. That's why I was saying, if you really want to disrupt the market and you really want to be able to help companies that are new to this field, you need to provide solutions that are quick, efficient, and economic. Otherwise, it's a daunting task, and no one will dare to get into it.
Yeah. How does it all compare when you think about, I always think sometimes, in Europe, we're a little bit under the radar, but if you think back to the billions spent on the likes of Ginkgo's foundry and the computational piece and all the kind of, what was Zymergen at the time as well. How quickly is this technology evolving, and how does your kind of tech stack kind of compare, as it were?
It's a different approach, right? Many of these companies have a very lab-intensive approach,
yes,
which works.
Right.
There's nothing wrong with these approaches. They work. It's just that they are super expensive. So where we excel, and not only ourselves. So think about it. We've been around for 8 years. When we started, the question was, what do you mean you can do this in the computer, right? Now it's more like, what makes you different from A, B, C, and D? There's no doubt anymore that computational simulations are helping in reducing the number of lab experiments. But for me, if you ask my honest opinion, I think the future is going to be a combination of both. If you really want to train these future AI models, you need to have a lot of experimental data. So these companies like Ginkgo, which are generating massive amounts of information, that's what companies like us, we need that. Okay, they might have the AI, but I think the physics base that we bring to the market, together with all of that data, I think that's the future. So these foundries and these capabilities to screen thousands, millions, I think they will always be needed. The thing is that they will be needed on specific projects, on the very difficult ones, or we will be able to speed up the development. So say instead of doing 2 or 3 rounds where we test 100 in each round, let's say we do one single round with a lot of data, but again, guided by the computer, right? So I think in the past 2 or 3 years, we've seen a real revolution in this field, and other areas like drug discovery that for decades now have been always screening everything in the computer before going to the lab. I think the same thing is going to happen to enzymes. I think that less than 2 years from now, there's not going to be a single enzyme development that hasn't passed by the computer first. Then sure, do as much lab work as you need, but do the heavy lifting in the computer so that what you send to the lab has really high chances of success.
Yeah, and you save a lot of time and money, no doubt, in that process.
Absolutely.
Yeah.
If the question initially was, can it be done? I can guarantee it can be done. It's been 8 years and a lot of projects and a lot of data. It can be done. Now it's just a question of how much more can we make this process efficient and economic so that it really changes the world.
Got it. Yeah. So what's your big vision for the company now in terms of, where things stand and where you are?
What we're working on right now, and it's a kind of there are 2 directions we're heading into. One is our own product development. So we have started a couple of years ago to develop our own product. So obviously we want to take advantage of our capabilities to bring new product to the market. So that's one of the big goals. And the second is to bring the market together. So there's already a lot of enzymes out there that work. You don't always need an optimized enzyme, okay? Sometimes you can just use something that's already available and just repurpose it for something else. But in order to do that, what companies are doing today is basically going to provider number 1, provider number 2, provider number 3, and just getting all of those commercial kits and testing them in-house. So our technology allows us to screen very efficiently. So why would you test 100 enzymes from A, 100 enzymes from B, 500 from C? If I can have all of those enzymes in our platform, we can screen it and we send to the customer, we say, test these 20. These are 20 commercially ready enzymes. So they're scaled up. If you want a kilogram of these enzymes, 2 weeks from now, they're available. This is how I see the future. ZYMVOL as the central hub of identifying any enzymes that are already in the market that already have been scaled, that we can repurpose them for new applications. Because then forget about 1 to 2 years of development, forget about the investment, forget about the risk. It's there. It's just a matter of finding them and sending them to the customer very quickly. So this is something new that we're developing right now, and it still hasn't been announced. But I really think that in the future, you need to have a ChatGPT for enzymes, and the answers are there. This is the enzyme you can order from this provider. Test it. If you're happy, order a kilogram, order a ton. It's all in the same place. So that's where I see our company in the future.
Great. And tell me about the product development side then, because that must be like, where do you start with that? I mean, it's industries, applications. I mean, I'd love to hear how you kind of approach that.
Good point. When 8 years ago I said, okay, let me set up a company, and I had to figure out everything that had to be done. It kind of feels like the same thing with this product. So this was a product actually that was discovered by one of our partners. They have been for many years our key provider in terms of all the lab work. This is a company in the Netherlands called Ginkgo. Ginkgo, not Ginkgo, Gecko. And basically they discovered, again, by chance, a product which is very, very interesting. It's called melanin. So the skin pigment that protects us from sun. This pigment today costs about 500,000 euros per kilogram. So very, very expensive, 5 to 7 times the price of gold, just to give you a reference. The thing is, melanin can not only protect you from sun, but there's also multiple applications from an ingredient that can be used in, of course, cosmetics, but also food, even in defense applications. It's got semiconducting properties, so it's a really awesome molecule. But you're absolutely right. For a computational chemistry company that has been developing enzymes for third parties, how do we bring this product to the market? What are the industries? What are the applications? How do we get this out there? It hasn't been easy. It has been maybe the biggest challenge we've faced so far. But we're right now ready to scale up the molecule. So we're going to start very soon scaling up production, because we still need to drop the production cost by still a few folds. But we know the market, we know the customers, we have letters of intent. So we're going to be scaling up our first product. And I have to tell you that we have 5 or 6 molecules in the pipeline, which are also in quite advanced stages of development. So yeah, developing product is a whole new story. But we believe melanin is a good molecules to start with. There's different opportunities there. I mentioned cosmetics, which is going to be our first target for sun protection factor. So this is the initial goal. We've already confirmed the characteristics of our molecules. So not only allows you to reduce considerably the use of other molecules, which will soon be removed from market. So that's a big advantage. But also our melanin is biodegradable. It's produced in a sustainable process. So, and it's also water soluble, which makes it also different from natural sources of melanin. So normally you take melanin from squid. You need to kill, poor squid. Take melanin.
Is that where it's from? I was going to ask
most
you
of the
where
time,
it's from.
yes. Or you produce it synthetically, and then the cost is 500,000 euros per kilogram. So it's not a simple molecule to come across.
No. I always remember. I can't remember what the molecule was, but Amyris had a particular molecule, and
is it a sugar?
Squalene.
Okay.
Squalene, and it used to be derived from, I think it was shark's fin or something like this.
Yeah, I don't remember the exact origin, but it's not the first time that, biotech is able to produce a molecule which is very difficult to source from natural and also bad for the environment, right? Because you're basically you're basically killing animals, or you're sometimes also it's the case you're interfering with the ecosystem. So there are many examples of biotechnology being able to produce molecules that are sourced from natural sources, but natural sources that you need to basically kill. So there's a huge advantage of through a simple fermentation process, just adding sugar and being able to produce molecules that otherwise are very difficult to come across.
Yeah, and so is that the case here? You start with like a yeast or something like this, and
we're producing our melanin with a bacteria. Currently, we will also be testing different oats, but we're producing it currently with a bacteria. And yes, and the starting point is some form of sugar.
Yeah. Well, it always makes great sense to me, just from being on the outside looking in, to start with a molecule which is did you say 500,000 euros a kilo?
Correct.
That's a good place to start, right? Yeah.
It is also one of the challenges of biotech, right? Obviously, the opportunities within health are clear because we're talking about massive margin markets. But when you're talking about synthesizing something, this is an instrument, right? You're just a catalysis for something that's really relevant for the market, but still, it's just an instrument to reach an end. So we need to make sure that when we're developing an enzyme, it needs to be producing something that's really relevant, because otherwise, likelihood that you're going to fail because you're going to be competing with other sources, and price is always going to be, money is king. If you can produce something, cheaper with a standard chemical synthesis, no one's really going to look at biocatalysis. It needs to have a clear economic advantage. Otherwise, it's not going to happen.
Yeah, absolutely. Which is the challenge, isn't it? That is the challenge. Tell me about other kind of markets or applications broadly. Not even the stuff that you're developing, but what you're excited about, because where you see kind of opportunities, as it were.
Well, novel materials, right? The materials of the future need to be thought of from the beginning until the end. You need to think about how to produce them, ideally from sources which are more accessible or better than fossil sources. Then you need to be able to recycle them back to the original molecules, right? So I think novel materials is going to be an area where there's going to be a lot of potential for biocatalysis. Agro has always been an area which is very interesting. So pharma is the obvious choice. It's been around for so long that there's always going to be opportunities there. But I think the most exciting for me would be probably materials, and also specific ingredients like melanin, ingredients that are versatile, that you can use in different industries, and then that you can produce in a sustainable way with enzymes. But I would probably say novel materials and recycling, like all the hype that we've seen, for instance, with PET, with recycling plastic back to the monomers. I think all of these complex polymers that need to be degraded back to their monomers is going to be, but already is, an exciting area to work in.
Yeah, absolutely. No, fascinating. It really is, and it sounds like it's been an education. You coming on the show today to have a kind of chat about this, as it were. And tell me, how many people work at the company?
We are 26 people right now, full time. We have a few advisors and people that work with us on a part-time, but we're 26 people.
Got it. Yeah, and I saw you just hired John Nichols, I think it was, right?
Correct. Well, John Nichols has been with us for about a year as an advisor, so he's been supporting us in the backstage for already quite a number of months. And he has been such an amazing addition to the team, particularly for me, because he's also becoming kind of my mentor. His experience as CEO at Codexis and his previous experience is just amazing to have him. But now he's also part of our board, and he's helping us particularly in the development of novel products, right? Because he's done this before, he's done this successfully before, and like you asked previously, this is not an easy adventure.
No.
Starting to put your own product in the market is definitely one of the most challenging moments for a company, but it's also one of the most exciting. So I think John is amazing. And we have also another person on our board, which is Pierre Monsa, who was the founder of Toulouse White Biotechnology. So he was already thinking about biocatalysis 30 years ago. And these two individuals on our board are the light that keeps us in the right direction. So I'm very, very fortunate to count on
Very
their
much
expertise.
so. Yeah, absolutely. I mean, yeah, I've worked with a few companies that have come out of the TWB over the years. So, yeah, no, aware of Pierre. So, yeah, very interesting addition. And look, just final couple of things, like from my viewpoint, how have you found the whole fundraising kind of process and that part of the kind of job?
It's a full-time job.
Yeah.
It's definitely a full-time job. It was very alien to me because as a scientist, this was, and especially after bootstrapping for 6 years, this was a new adventure. We've been very fortunate. So we've always been able to select the funds that have invested in our company. The first fund was Elia Partners that invested in 2023. Single investment. That was one of the conditions we had for pre-seed. We wanted to have a single fund. And now very recently we've raised with still Elia, Favor Ventures, a fund from Portugal that led the process. Morgan from Switzerland, and a small fund here from Spain called Macia. So we were very fortunate because these are impact funds, deeptech funds, thinking about how can we change the world. So they keep us also grounded in, okay, I know you have your own mission, you want to expand biocatalysis to the whole world, but this is more about just biocatalysis. This is about changing the world. And I think we've been very, very fortunate. It's a tough moment to raise for biotech right now. We were oversubscribed in the previous round, which I think is, very fortunate in the current environment, but we're still going to keep on pushing forward. And right now we're very well surrounded with the funds that have invested in ZYMVOL.
Oh, that's awesome. I was going to say, do you see, like, it has been a real challenging kind of for biotech at the moment, and hopefully we're past that worst though, and I think we just need a few quick wins out there, or a few kind of real substantial kind of wins.
Yeah, I think we need to see products in the market, and we need also, and I've been saying this for many years, we need to truly understand that synthetic biology, biotech, if you exclude, all the health-related applications, these are instruments, these are tools to reach a specific goal. Investing billions into a tool is not likely to happen. So we need to understand, at least in my view, and again, this is very personal, I think the opportunity is building many such tools and not just having that one blockbuster that is going to change everything. So we need to invest wisely also in biotech and make sure that we put our bets on things that are likely to succeed.
Awesome. Yeah. Listen, I could chat to you all day. You're so easy to talk to. And I really appreciate you coming on. We've learned loads today. I mean, you've really given a fascinating kind of, description about, what you do and your technology and how it differentiates, and just your vision and your view of what you want to accomplish, and just your personal journey as well has been really interesting. So thanks so much for coming on and sharing today. Really enjoyed chatting with you.
Thank you, Simon. It was really a pleasure, and I'm here anytime you want to talk about biotech.
Awesome. And look, to all our listeners, thanks so much for tuning in. I'm sure you'll really kind of enjoy the this week's show, and we'll see you on the next one.